In this part of the video, we will be talking about the next chapter, that is the transport of substances through the cell membrane. Here, first we will discuss a little bit about the cell membrane, then the diffusion, facilitated diffusion, and the active transport. In this particular video, there will be two topics, diffusion, facilitated diffusion, and one more topic, that is osmolarity and osmosis. First, let's know or let's revise about the cell membrane, that they have lipid bilayer, that is inside they are hydrophobic, and outside they are hydrophilic. There are presence of channel proteins, which we have already known. There is presence of transport proteins. These are channel proteins and carrier proteins. With the help of channel proteins, there will be simple diffusion, and with the help of carrier proteins, there will be facilitated diffusion, or if it requires more energy like ATP, it will be active transport. So diffusion can be of two types, simple and facilitated diffusion. In simple diffusion, there is no requirement of any carrier, but in facilitated diffusion, there will be requirement of a carrier. So few points about cell membrane. So differentiating between diffusion and normal active transport, in diffusion, there is a particular or molecule movement in between the intermolecular space or with the help of carrier, like for facilitated diffusion. But in active, it happens in the same fashion, but it requires more kinetic energy. So here, it goes from low concentration to high concentration in the active, whereas in diffusion, it goes from high to low concentration and requires less kinetic energy over here. The next is diffusion. So diffusion can be of two types, that is facilitated and the simple diffusion. There are several factors which defines them. For example, the first we have the lipid solubility. We know that the cell membrane is made up of lipid bilayer. So first of all, some lipid soluble factors will go through the cell membrane itself. So if the solubility in lipid is very high, that means they can cross the cell membrane very easily. For example, we have oxygen, nitrogen, and carbon dioxide. That means they are directly proportional to the lipid solubility. So diffusion is directly proportional to the lipid solubility. Since water cannot pass through the cell membrane because there is lipid bilayer, so there is presence of protein channels, which we have discussed. That is the transport proteins, that is carrier, and the channel proteins. So these are termed as the protein channels. These protein channels have pores. The pores for water is called the aquaporins. The aquaporins will only allow the water molecules to pass through it. Hence, it suggests that it is selectively permeable. And the other factor about that they can be open or closed gate. So the first is selective permeability. For example, if we have sodium channels, so only sodium will pass through it. If we have potassium channels, only potassium will pass through it. If calcium channels, then only calcium will pass through it. No other molecules can pass through them. So that is, these are selectively permeable. Specifically to the sodium channels, they are zero point 0.3 to zero point 0.5 nanometers in size. The next is the open or closed gates. These are regulated by either by voltage or by the chemical ligand. So in case of voltage gates, there is presence of negative or positive charges. For example, when the sodium is trying to enter, there is negative charge because sodium is present mostly in outside of the cell. So now it has to enter from outside to the inside. Hence, there is negative charge present. For example, in potassium, but in case of potassium, there will be presence of positive charge, and all of them will be open at the same time or closed at the same time. It does not happen that one is open and one is closed, one is open and another is closed. No, all of them will be open or they all will be closed. Let's say here is one channel. Now this is outside and this is inside. Now here is negative charge present. Then a sodium molecule is coming here. These negative charge will be released from inside first, then there will be opening on this side, and hence slowly the sodium will come inside. Within a fraction of second, the sodium will come in. These are necessary for nerve signaling. Signaling. These are necessary for nerve signaling in the nerve cells. Then the next is chemical ligand or the chemical gates. These are activated only when a chemical substance is attached to them. For example, we have acetylcholine receptor. When acetylcholine is attached to the surface of these proteins, then there will be the opening of these gates, and transmission of nerve signals can pass through from nerve to the muscle cells for the muscle contraction. The next is the facilitated diffusion. In facilitated diffusion, the maximum speed is reached always, whereas in simple diffusion the velocity will be always increasing. For example, there are several molecules present, then the velocity of each and every molecules will be taken into account. Whereas in facilitated diffusion, whatever be the quantity of the molecules, they will reach a maximum velocity. That is, the rate of diffusion will reach a maximum. It is because usually there is a binding factor which is very weak in nature, and there is thermal regulation also happening, which leads to the breakage of these molecules inside the cell or outside the cell if it has to go. So the binding chemical nature here is very weak, and the thermal binding is also very sharp. The factors influencing the diffusion is: number one, the rate at which outside concentration and the inside concentration. The O represents the outside and I represents the inside. If there is more concentration on the outside of the cell, then it will be diffused from outside to inside. For example, just in case if there is more concentration on the inside, the here this will be turned from Ci to minus Co, because the concentration in the inside of the cell is more and now it has to go outside. The second is the pressure. That is, they go from high to low pressure. It happens because here, let's imagine here a cell or a wall. These are the molecules, and here are less molecules. So each of these molecules each of these molecules will apply pressure on this wall. So because of such pressure, they will diffuse from one side to the other side, that is high to the low pressure. And this is in case of the capillary, that is inside it is greater than 20 mmHg than the inside. And the last is the electrical changes in the cell. For example, we have already seen in case of sodium and potassium, if the are closed, sodium gates are closed at the starting, they will not be able to enter, even though there is no concentration gradient, but because the negative charge is moving away within the inside of the cell, hence the gate is opening at the outside, and which leads to the intake of sodium. So there is no concentration, again the electrical changes in the cell is happening. Also in the facilitated diffusion, the examples could be the glucose, that is with the help of GLUT4, that is the carrier for the glucose, and several amino acids. And there are more examples. These are the basic examples from which these are taken away by the process of facilitated diffusion. The next is osmosis. Osmosis is a special name given for water. There is no difference, it is going from high to low concentration as well. That is, osmosis happens when there is a concentration difference. Usually there is no concentration difference, and hence the osmosis does not take place. So when there is concentration difference, the osmosis takes place. That is, the water goes from high concentration to low concentration. Now, to understand osmotic pressure, we have one experiment of U tube. Now, in an experiment, to understand this, in an experiment of U tube, here is a semi-permeable membrane. Now the water has been filled over here, and there is little water over here. And this is chamber A and this is chamber B. There are particles present over here. This space is causing the osmotic pressure over here. How we understand is the water has moved from chamber B to chamber A. That is, first here was the higher concentration. In chamber B, there was higher concentration, and then later on, where let's say here is sugar, the water will go into the A chamber, maintaining the concentration over here. That is, this pressure which has led to the space between from here to here, and here in chamber A it is somewhere up. So this is the osmotic pressure over here. This is causing, it is actually the amount of pressure required to stop osmosis. That is called the osmotic pressure, because after this pressure has been created, there will be no further osmosis going on. That is, the water will not diffuse from a high concentration area to its low concentration. So it is measured in molar concentration, that the number of particles per unit volume of fluid exerts pressure, not the mass of the particles. So if there are more particles present, that is, the pressure is more. If there is less particles, again there will be, the pressure is less. So difference between osmolarity and osmolality is that the osmolarity with R, it is expressed in terms of per liter of solution, whereas osmolality is the same thing, the osmolar concentration, but it is expressed per kilogram of water. So the normal for intracellular and extracellular fluid is 300 millimoles per kg of water. In the next video, we will be talking about the active transport.